Assalamu alaikum, dear students. How are you? Students, how are you? So today is our class. I was thinking that today is not our class. I I was thinking that tomorrow and day after tomorrow will be continuous classes. So it was my mistake to rector it. That's why we started late. But no problem. We are starting it again. So where were we yesterday? Saad. Students, where were we yesterday? Students who says cell division. We yesterday. What is cell division? Who will tell me the cell division? What is cell division? Muhammad Mohid. Mohid, would you please tell me what is cell division? When a single cell divides into daughter cells is called cell division. Mohit, you are right, but when the single cell divides into two daughter cells, not only single daughter cell, just into two daughter cells is known as a cell division. Okay, so this must be understood that cell division is the division when the cell divides into two or more than two daughter cells is known as a cell division. Okay, tell me what are the uh, how many types of cell division are? How many types? Two types, Mohammed Wahid? Okay. Anybody else? Okay, okay. Anybody else? Hunain said there are four types of cell division. Okay. Okay, there is a poll and it asks you that how many types of cell division are. Answer the question. Yes, Osman. Answer the question. How many types of cell division are? Students, are you there? Only two has responded. Where is Saad? Saad and Hunain should respond me. Sorry, sir, I don't recall. Mohammed Saad says, why, Mohammed Saad? We studied yesterday about the divisions, types of divisions of uh, cell, among which we studied one type and other types are remaining. So, how many types of cell division are? Students, tell me how many types of cell division are. Okay, it has three types of cell division, not two, but three types. Which I told you that it has three types of cell division. One is mitosis, other is meiosis, and last one is binary fission, which occurs in the bacteria or prokaryotes. Okay, did you recall it now? So today we studied. Is it clear now? I think it's a network problem. That's why it's going on. Otherwise, we are going to continue it. Okay. So what is binary fission? Students, can anybody tell me that what is binary fission? In which organism it occurs? Binary fission occurs in. Very good. Very good, Raza. Very good. Today we are going to learn about the mitosis, and as we studied yesterday that what is the cell division, we also studied the types of the cell division and why you want to reconnect. I think there is a network problem also. Okay, so today we are going to learn about the mitosis. What is mitosis? Mitosis is a form of cell division or type of cell division by which the eukaryotic somatic cells duplicate. Yesterday I told you about the somatic cells. Do you remember the somatic cells? The somatic cells are the body cells. So in eukaryotic cells, the somatic cell and eukaryotic somatic cells, the cell division occurs. This is known as a mitosis. Mitosis is an asexual reproduction. Is a type of asexual reproduction, which I told you that yesterday that in these somatic cells, in the body cells, it is the cell is dividing for the growth purpose. Otherwise, the germ cells, meiosis occurs in the germ cells, which we will study in day after tomorrow. So cell division is a continuation of life based on the reproduction of the cells. Okay. So mitosis, how mitosis is working out? You can see here that we in humans, the liver cell or all the body cells consist of the 46 number of chromosomes, and these 46 number of chromosomes can be denoted as a 2n. It means they are in a diploid number. Which number? Diploid number. Diploid number. What does it mean, the diploid number? The diploid number means that it is the number of the two copies of the same chromosomes. It means we have the 23 pair of chromosomes only. Okay, so in our body only 23 pairs of chromosomes present. It means we have 23 chromosomes and their 23 copies are present. It means it has 2n number of chromosomes are present. They are known as a 46 chromosomes, and they when they divide, they again divide into the two daughter cells, and both daughter cells have the same number of chromosomes. So mitosis, when divide, the mitosis cell, when the cell divides with the help of mitosis, it means the cell produces its daughter cells with the same number of chromosomes as their parents or mother cell had. This is the mother cell, and these are the two daughter cells. It means the two daughter cells can be formed without any difference in the chromosome number. So chromosome number in mother cell will transfer the same number of chromosome in their two daughter cells. So you must remember that chromosome number will be same in the mitosis. So somatic reproduction, most eukaryotic cells reproduce asexually by the mitosis. The somatic cells are all the body cells like nerve cells, liver cells, stomach cells, muscle cells are the somatic cells, except the sperm and egg. Sperm and egg are not the somatic cells, but they are known as the sex cells or the germ cells. All the somatic cells have the same number of chromosomes. You must remember that all the somatic cells have the same number of chromosomes, while sperm and egg have the half number of chromosomes. It means they will have the 23 number of chromosomes, while the somatic cells have the 46 number of chromosomes. Okay, the what is the cell cycle we studied yesterday, but we are again going to revise it, that what is the cell cycle. The dividing and non-dividing stages of the cell. Phases are the interphase, prophase, metaphase, anaphase, and telophase. Interphase stage is a growth and DNA replication stage, and while the somatic stage, while the other mitotic stage consists of the prophase, metaphase, and telophase stage, which we will study in this diagram, which I told you yesterday. It has the four type of phases: G1 phase, S phase, G2 phase, and M phase. And mitosis phase consists the subphases, which are known as a prophase, metaphase, anaphase, and telophase. So now today we are going to study about the mitosis. We have studied the G1 phase, S phase, and G2 phase yesterday. Now we are going to study the mitosis in very much detail because your course outline is that. Okay, so we are going to start that interphase stage and the mitosis stage. But before going to that stage, I just want to go for the overview about the interphase stage, which consists of the S phase, G1 phase, S phase, and M phase. Oh, sorry, the interphase stage consists of the G1 phase, S phase, and G2 phase. These three phases are combinedly known as a interphase, and other stage is known as a, this part is known as a mitosis phase or the M phase. Okay, so we are now going to overview for the interphase. It comprises about the 90% of the cell cycle. Cellular growth occurs in this interphase stage where the protein synthesis, metabolic activities, DNA synthesis are all these processes occurring in the interphase stages. As we studied yesterday, it is made up of three phases: G1, S phase, and G phase, G2 phase. G1 phase is responsible for the protein synthesis and metabolic activities. Most cells are arrested in this phase. S phase or the DNA replication takes place. G2 phase, cellular growth and preparation of the M phase takes place. So there is all these things are occurring in this phases, interphase stages. In the interphase stage, the nucleus and nucleolus are visible, where the chromatin and nuclear membrane, nucleolus and cell membrane, you can see here that this is the typical structure of the cell, in which this is the chromatin material in the gray part, this is the nucleolus, and this is a nuclear membrane, and this is a cell membrane. So question arises here, what is the chromosome? Chromosome is a chromosome is made up of DNA and histone proteins, and they make a complex called the chromatin. It means the chromatin is the combination of the DNA and histone proteins. When both combine with each other, they form the chromosome. So chromatin is a long, thin fiber that is folded and coiled to form the chromosome. Okay, here you can see that this is the chromosome. When we unfold the chromosome, it forms the thread-like structure. In thread-like structure, if we more straight it out, we see that these black dots with the thread-like structure, these black dots are the histone proteins, which are combined with the chromosomes or this thread-like material. When this thread-like material again uh, opened up, it shows the double helix of the DNA. And when it again be opened, it will show us the genes of the cell. Okay, so all these things, all these chromosomes or chromatin material is present inside this nucleus with in these form of the threads. So you must remember that this is the animal cell. In animal cell, this is the nucleus, and inside the nucleus, these are the thread-like material which are known as the chromosomes. When these chromosomes are opened up, they show up the histone proteins also and the DNA are double helix, and these two histone proteins and double helix combine to form the chromosomes. So what is the replicated chromosome? A replicated chromosome consists of two strands of identical chromosomal material called the chromatids, which is known as a sister chromatid. Now we can say that this is the replicated chromosome, this simple chromosome, and this is also the chromosome, but this one is a chromatid and this one is also a chromatid. This single whole part will be known as a chromatid. Now this single part is a chromatid and this is also the chromatid. So that's why both chromatids will be called as the sister chromatids. And this is the one chromatid, and when the S phase occurs during the S phase, chromosomes replicate and it forms the same copy of this chromosome in the form of this, and these two copies are joined together with the help of a particular type of protein which is known as a centromere. So centromere is a type of protein which combines the two sister chromatids with each other. So this is a single chromosome and it makes its own copy of the same type of chromosome, and that copy is joined with the its original chromosome with the help of centromere, and then now this is known as a chromatids, and this is also the chromatids. Now these both will be known as a sister chromatids, and these two sister chromatids combinedly known is the as a chromosome. You must remember that this is a single chromosome. If it replicates in the S phase to form another copy with the help of centromere, it combines both of the sister chromatids with each other and makes the complete chromosome. A diploid chromosome. You can see here, this is the diploid chromosome. You can call it the 2N chromosome. Okay. Now we are coming towards the mitotic phase. What is yesterday we studied there are two phases of the mitosis. One is karyokinesis and other one is a cytokinesis. In karyokinesis we can see that division of the nucleus takes place, while in the cytokinesis division of the cytoplasm takes place. So in the nuclear division the genetic material also replicates to form the divide to help in the division, and prophase, metaphase, anaphase, telophase stages are the part of this karyokinesis. While in the cytoplasmic division, it indicates the indicator of the telophase stage shows that the cell is completely divided into the two daughter cells. Now we are coming towards the prophase stage, the first stage of the mitosis karyokinesis stage, and that is the longest phase where the nucleolus disappears. Chromatin material chromatin material condenses into the chromosomes consisting of the sister chromatids. In prophase, centrioles move apart, not found in the plants. These centrioles are not found in the plants, but in the animal cells they move apart in the side in the spinal formation. The spinal forms these are the types of microtubules which are being formed for the purpose of the attachment of the chromosomes from the where the centrioles they attach to the centromere and by the kinetochores. Nuclear envelope fragments and disappears. So these are the things which happens in the prophase stage. Number one stage there is nucleolus. In the prophase stage we see this nucleolus disappears. First of all, what happens during the prophase? Nucleolus disappears. When the nucleolus disappears, what will happen? The centrioles, the cell organelles which we studied in previous week are in the animal cells move apart. There are two centrioles, they move apart, they separate from each other, and they one goes the one part of the nucleus and the other one goes the other part of the nucleus, from where it is spread or emits the new microtubules are known as a spinal fibers which connect which connect the centrioles with the centromere, which we studied the centromere, which is a part of the two chromatids between them, which connect the two chromatids, sister chromatids with each other with the help of kinetochore. Kinetochore is another type of protein which combines the centriole with the centromere. So nuclear envelope and breaks down and disappears slightly. So there is no any nuclear membrane now during the prophase stage. There are three steps are happening inside in the prophase stage. Number one, there is nucleolus disappearing. Other one, centrioles move apart and combines with the centromere with the help of kinetochore. And number third point is the nuclear membrane disappears completely. So there are three parts of the prophase. Here you can observe these three type things which we will we can see this is the early prophase where you can see these are the centrioles. This is the nuclear chromatin material, and this is the we can say this cell membrane. But after the phase, we can see here that this centriole is moving apart from one side, and the other part will be moved from here side, from this side and this side, and these are the chromosomes. And in the chromosomes, this is the centromere attached here with, and you can see there is no any nuclear envelope available outside the chromosomes. So this is the late prophase when the nuclear membrane disappears. Nucleolus, you cannot see here the nucleolus. There is centrioles, which are one part is here, another will be here. This picture is taken from the original cell division, and it is in from the through the electron microscope. Okay. Now we are there is another question which asks I think there is a network problem, therefore the slides are not coming very much early. Okay, student, do you see the question? What attaches the spindle fibers to the centromere? I told you before this slide. Students, can you answer me this? What attaches the spindle fiber with the centromere? I can ask you the questions also. Question also. Number one. Chromatid. This is a question for you. Can you answer this question? What attaches the spindle fiber with the centromere? Students, answer the question. Saad, answer in the poll. Students, Usman and Saad, answer the question. Its answer is kinetochore. Okay, let me show you the previous slide. See here, this is the kinetochore. The centrioles are connected with the centromere with the help of kinetochore. Do you see this? Is it okay now? Students, you can see a kinetochores. This is the answer. The This is the central, and this is the centromere. And in the centromere, this is a small part which is known as a kinetochore, where these spinal fibers are connected with this kinetochore of the two sister chromatins. Okay, Hunan, doesn't matter. Sometimes it happens that there's a network problem. Okay, so you can, Hunan, you can get the video downloaded after this lecture also. My videos are available on there. If you anybody wants to listen the videos again, they can go for the videos and can download it. If they can't find, I can give the link to you to download or watch the videos again. Okay. Okay, students. So this is the metaphase stage. Before we studied the prophase, then metaphase stage, the shortage phase. Centrioles are at opposite end of the cell and attached with the ester fibers. And centrosomes move to the metaphase plate, equatorial plate, and center of the cell. Now you can see here, this is the centriole and this is the centriole. Now these chromosomes are at the center, and these are the ester fibers which are connecting with the kinetochore, each chromosome's kinetochore, and at the equatorial sides. And this is the spindle fiber. Okay. In metaphase stage, top of the spiralization chromatin is a maximum of the condensation. Chromosomes collect at equatorial plate. At the end of the metaphase, centromere start disappears. Okay, so this is the centromeres will be start disappear. In the anaphase, centromeres uncouple and spinal fibers shorten. Sister chromatids separate and move apart. And after separation, chromatids are now considered as a chromosomes. And in anaphase, during this phase, the cell contains twice the normal number of chromosomes. Cell begins to elongate. At the end, there are equal numbers of chromosomes at the poles. Okay, you can see here. Now these chromosomes were attached previously in metaphase stage. Now these spinal fibers are going to be shortened, and one part of the sister chromatid is going this side, and one part of the sister chromatid is going this side. And then what will happen? These two fibers are joining together in the side by side, the anaphase stage. Okay, so in telophase stage, nuclear membranes reappears, chromosomes uncoil and decondense, and at the end, two genetically identical nuclei, karyokinesis completed, are present. In the picture, we can see here that you can see this all the chromosomes are presented here, and these are the here. I am sharing you the main points, Mamunwar. This is only the main points. I'm not going in detail because of the short time period. Okay, so you can see here, and we are going to the cytokinesis. In the cytokinesis, you can see here these two daughter cells are being formed, and okay, so please share the main points PPT. Okay, this is our end of the topic, and this is the cytokinesis. I will share this PPT with you. You can read this, and you can also understand of this lecture. This lecture in detail we will study tomorrow, and for the meiosis also. Okay, so day after tomorrow. Sorry, tomorrow is not your class. Is it okay? Tomorrow is not your class. Day after tomorrow is your class. Okay, so students, this is our class, end of our class. Tomorrow, day after tomorrow, we will again study. So see you tomorrow. Hafiz to tomorrow.